Thank you. Yeah, thanks very much for inviting me to this great conference. It's my first time being to the Allen Institute. As you can see here, I'm just over the water at the University of Victoria. So the stuff I'm going to talk to you today about is basically one project going on in the lab, trying to explore the possibility of using optogenetics to enhance a recovery from stroke. So as most of you are aware, stroke is generally considered and comes in two flavors. You have the ischemic type stroke, which consists of about 85%, the majority of strokes, and the other remaining 15% is hemorrhagic. So stroke is obviously a big problem in North America and around the world because it happens quite frequently. But the big problem with stroke is that the majority of people that have a stroke will survive it, but they'll also have long-term deficits and disabilities. For example, I think this timer is going on. So they'll have long-term deficits in the ability to use their hand, long-term abilities in using the arm, and so things you take for granted, like using a fork or brushing your teeth, are impaired long-term, which obviously affects your independence in daily life. And we know that even 6 months later after an ischemic stroke, many of these deficits persist. So hemiparesis is still present in about 50% of people long-term after stroke. And so you can see here that we've been getting better at treating stroke in the acute short term in the first couple hours after stroke, getting people to the hospital, which has led to increased survivability. But the problem is we have more people living with disability. So in my lab, we try to model stroke impairments in mice. So this is modified from a paper of ours from a couple years ago. And so you can see here in using these simple behavioral tasks, here's the adhesive tape removal task where you put tape on each paw and look how long it takes the mice to take the tape off, that the impaired paw, the left paw here, shows these deficits immediately after stroke, and that can persist even out to 10 weeks relative to the unimpaired paw. And similarly, when we're looking at how a mouse walks across a ladder, they can have these persistent deficits in how they place their paws on the ladder, which we can quantify. And for example, in this here, you show the correct placements is around 75–80%. That drops immediately after stroke, and there's a bit of recovery over time, but nowhere back to baseline. Okay, so in the field of stroke, this is work from, some of this is my work when I was a postdoc in Tim Murphy's lab, looking at what happens to the brain after stroke and what correlates with these behavioral deficits. And so what we did in this paper back in 2009 is we used voltage-sensitive dye imaging to image the cortical representation of the forepaw, the stroke-affected forepaw, and looking at how that responsiveness to forepaw touch changes after a stroke. And so before stroke, you have a robust response in the forelimb somatosensory cortex. 1 week later, these responses are greatly diminished. And then 8 weeks later, there's a restoration of responses, although not nearly to what it normally looks like. And similarly, we've seen this with intrinsic signal optical imaging or single-cell calcium imaging right after a stroke, where the cortex just isn't very responsive. One of the ideas behind this loss of responsiveness after stroke in the cortex is that there's changes in intracortical circuits that could underlie this. But one thing that hasn't been checked much at all is what might be happening to circuits upstream, for example, from the thalamus to the cortex that bring the sensory information into the cortex. So just to refresh everyone's memory of the circuitry in the somatosensory system, so you've got proprioceptive or mechanoreceptive receptors that sense joint position or tactile stimuli. It gets relayed to the medulla, thalamus, and thalamus to the cortex. And so we really don't know what's happening to these connections after a focal ischemic event. What we do know from histology as well as in vivo two-photon imaging, so if I inject an AAV bilaterally into the thalamus, into the somatosensory nuclei of the thalamus, you can see there's robust labeling in layers 4 and particularly layer 1 as well. And then after stroke, there are residual thalamic axons next to the stroke in the peri-infarct regions, which is where we see this remapping of cortical functions after a stroke. So one of the first things we want to do is ask us, are these connections that survive, are they working properly? So an experiment we did is we used these genetically encoded calcium indicators, so GCAMP6S, and injected that into the somatosensory nucleus of the thalamus, into the VPL. And then we'd put a cranial window over the mouse's head, and then we'd use intrinsic signal optical imaging to map the forelimb and hindlimb somatosensory representations. And then what we did is we, using two-photon imaging of the GCAMP labeled thalamocortical terminals in layer 1 of the cortex, we'd image their response to touch before stroke and then 7, 14, 21, and 28 days after stroke to see how stroke was affecting these viable projections. And so this is a map. On the right is a delta F heat map. On the left is the fluorescence change. So you can see these arrows are when we touch the forepaw at time 0 using 100 hertz stimuli for 1.5 seconds, you can see that you get a bunch of boutons that flash, respond to the sensory stimulus. And then, so we did that before stroke and after stroke. And you can see we focused on these peri-infarct regions next to the focal stroke here. So 7 days after stroke, when we touched the forepaw, there was very little responses to touch, as you can see by the cool colors here. Then we look 14 days later. Some boutons started to respond. So if you've got the boutons labeled 5, 6, 7, 8, 9. So bouton 8 here showed a good response in red here. You see it's locked with the sensory stimulus. And in some cases, we were actually able to image the exact same boutons 2 weeks later in the peri-infarct cortex. And so this bouton 8 is still responding to touch. But some of these other boutons that didn't respond 2 weeks earlier are now responding to touch, as you can see by these flashes of color next to them. And so if we just quantify the percentage of boutons that respond to sensory stimulus before stroke, it's around 25%. And then that drops precipitously in the first week. And there's a little bit of recovery from 2 to 4 weeks' time. So we know from this GCAMP imaging experiments that structurally these axons are still there, but they're just not responding very well to, or they're not delivering tactile stimuli, sensory information to the cortex through the thalamus. So one obvious suggestion is to use optogenetics to try and drive responsiveness of these projections after stroke. And so in our study, we used the second generation E123A variant of the channel rhodopsin protein. And one of the reasons we did this approach was, so there is a study that came out 2 years ago in PNAS from Gary Steinberg's lab showing that, and what they did is they optically stimulated layer 5 output projection neurons after a stroke, and they did it for 1 week after a stroke, and found a transient benefit in paw function and sensori-motor function that disappeared at the end of the study, but they only looked about 10 to 14 days after a stroke. But as stroke survivors all know, recovery takes a long time, takes weeks and months to occur. And so one of the things we did differently in this particular study is we thought of using a non-invasive way generally non-invasive way of optically activating these thalamocortical connections by putting a surface LED hat, basically with this magnet, which would then attach to the metal ring here, and then we could deliver stimulation through the cranial window, and then we could also image structure, for example, of thalamocortical connections using two-photon microscopy with this window. And we could do a sort of a high-intensity regime. At least what we did is we'd stimulate for 5 hertz for 1 second, 4 seconds off, 5 hertz, and then 4 seconds off. Basically did this for an hour a day for up to 6 weeks after a stroke. And so I've just kind of described those parameters here. There's our mouse with his window. There's the little head cap. And so the power densities we used are around 10 milliwatts per millimeter squared. Similar power densities that Carl Svoboda and associates have used in previous studies using this LED hat approach. And so you can see here in this particular experiment, we injected the channel rhodopsin into the ventral posterior lateral nucleus of the thalamus. And we'd also inject another AAV to drive expression of M-cherry, so we could look at the structure of these axons over time as we're giving this optical therapy. And so you can see here the expression in the forelimb somatosensory cortex. And then we do 2-photon imaging of the thalamocortical axons before a stroke and then for several weeks after a stroke, as well as correlate these structural changes with behavioral changes in mice that receive the therapy. So just to summarize some of the proof of concept experiments, we did some field recordings in layer 2-3 of the somatosensory cortex in response to a mechanical deflection of the paw, the contralateral paw. So we got these little deflections, and then we did different power densities of the LED light to try and see which sort of power density would elicit a response that would be somewhat comparable to the forelimb mechanical stimulus. And so ultimately, and then you can wipe these out with glutamate receptor blockers, but we settled on 10 milliwatts per millimeter squared, which was somewhat comparable to the type of cortical response in layer 2-3 that we'd get with a 4-paw tap. And so here you can just see the cortical potentials, field potential responses with 5 hertz tap versus the 5 hertz blue light stimulus at 10 milliwatts per millimeter squared. One thing we wanted to check first was, were we affecting blood flow? So we know we can drive cortical responses optically, but were we affecting blood flow? So we used laser speckle contrast imaging to try to reveal whether there was any change in blood flow with this hour of periodic optical stimulation. And so this is our baseline before we do that hour of stimulation. Here's 1 hour later. And you can see in the graph here, if we check the laser contrast, there's no change with optical stimulation or no stimulation, or if we just gave light stimulation without the channel rhodopsin. So we had different controls. And basically dark is these are flowing when vessels. And then just to prove that when there's no change, we did a positive control. So we did hypercapnia. So 90 seconds of hypercapnia. You can see that everything gets darker, meaning that blood flow is increasing uniformly in the cortex under a hypercapnia-like stimulus, but not when we did our 60 minutes of periodic optical stimulation. Okay, so this is just a little movie showing from underneath what the mice are doing. So we do this when the mice are in sort of just a home cage, freely moving. We just have the beak here so you can see them from underneath. We didn't see any obviously abnormal behaviors when they were getting stimulated with the light. So one of the first things we looked at is how is this optical stimulation affecting structure of thalamocortical boutons. So these boutons, or these axons, have en passant boutons. So these varicosities here are called en passant boutons. And then they have these little lollipops sticking out of them, which are called termino boutons. And so if you just look at the fraction of boutons as a function of these two types, the majority are the en passant variety, but there are some, about 30% of termino boutons. And so first looking at these termino boutons, if I can just show you an example here of here's a termino bouton before a stroke, and then 1 week later you can see this guy's retracted. So if you look at turnover rates, elimination rates of these boutons, you can see it's fairly steady, around 10% in both groups. It rises after stroke, and then the optically stimulated guys here in teal show a little more bouton elimination for up to 5 weeks after stroke. Similarly, the bouton formation in the optically stimulated guys was significantly greater than the control-stimulated guys. And controls, again, refers to mice that had channel rhodopsin injected but didn't get light stimulation, or got light stimulation without the channel rhodopsin. Okay, so how about en passant boutons, and what was the effect of stimulation on those, which represent the majority of thalamocortical boutons? And we know from serial section electron microscopy that if a bouton is about 2 times the median fluorescence of the backbone or the shaft of the axon, that when you do serial section EM, these have all the hallmarks of a synapse. So we know from at least ultrastructural data that these do have actual synapses. And you can see by doing longitudinal 2-photon imaging of these en passant boutons before stroke, the blue are the stable boutons that are present for all the way up to 4 weeks after the stroke. The red ones are ones that were eliminated. So you can see here in the first week after stroke, several of these boutons are disappear. But then there's some progressive addition of boutons over that month of recovery. So how did the optical stimulation affect that parameter? So if we look at the elimination of boutons, there wasn't a much difference between the groups. They both showed an increased elimination of boutons in the first week after stroke, and then it was somewhat variable for the 5 weeks. But if you look at formation of boutons, you can see that especially 2 and 3 weeks after stroke, there was an enhanced formation of en passant boutons in our mice that received the chronic optogenetic stimulation. My postdoc then broke down the boutons did this analysis of looking, and reminiscent of Kurt Haas' work a couple of years ago, looking at boutons that were pre-existing, so they survived the stroke, and then looking at their survival 5 weeks later or 4 weeks later, versus boutons that formed 1 or 2 weeks after a stroke, so they would be during that period of optical stimulation that would commence at 3 days and go out to 6 weeks. And so if you look at the pre-existing ones, so these are the stable boutons, their survival wasn't affected by the optical stimulation. And now if we look at the new boutons formed 1 or 2 weeks after a stroke, and we could subdivide it into some of the boutons that were expressing channelrhodopsin or not expressing channelrhodopsin, but these both would have received the optical stimulation. We can see that the channelrhodopsin stimulation enhances the survivability of these newly formed boutons up to 5 weeks after a stroke relative to a control group. Okay, so this is nice that it affects the structure, promotes the formation of new thalamocortical boutons that tend to be more stable. What effect does it have on sort of the network as a whole? So we used intrinsic signal optical imaging here to map the forelimb cortex before and after stroke. So this is the forelimb representation in both the control and the stimulated guys. We target the forelimb area, and then usually there will be a little piece of cortex left over that you can see gets activated when we stimulate the paw. But you can see here when you compare this group to this, or this mouse to this mouse, is that the area of cortex that becomes activated or recruited by sensory stimulation after stroke is enhanced in the stimulated guy as well as the magnitude of the response. So this is the area of response, drops after stroke, and then increases significantly in the stimulated versus the control. And the peak response as well drops after stroke, but starts to climb higher in the stimulated versus the control guys. And this goes all the way out to 10 weeks. And I should note that there were no significant differences between groups 2 days after the stroke before the stimulation started on day 3. So they're kind of starting from the same level of impairment. So ultimately, does this have any effect on behavior? So we used the ladder walking task to assay paw placements before and after a stroke. And so if you just focus on the correct placements where the paw makes a grasp centered over the rung, before stroke they're equivalent. After a stroke, 2 days later, both groups are equivalent, but then they start receiving the stimulation 3 days up to 6 weeks. And you can see that the stimulated guys accelerated the recovery of the percentage of correct paw placements on this task. And vice versa, the number of incorrect placements was significantly reduced in the stimulated guys. And similarly, in the tape test, which just looks at asymmetry and paw attention, the stimulated guys were equivalently impaired and then showed more equal symmetry between the stroke-affected and unaffected paw versus the control guys. And this persisted even after the stimulation had ceased. So this is my last data slide. Basically, the differences cannot be explained by any difference in infarct size. So optical stimulation starting 3 days after stroke doesn't make the stroke appear smaller or prevent ischemic death. It seems to be optimizing the function of the circuits that survive the stroke in the periinfarct cortex. So to summarize, we can enhance the stimulation of new and stable thalamocortical synapses with optical channelrhodopsin stimulation. It improves the recovery of cortical responses to touch that we showed with intrinsic signal imaging, and accelerates the recovery of sensory-motor function with the behavioral tests that we looked at. So just to thank the people in my lab and the funding agencies. Kelly Tennant is the postdoc that did most of these experiments, and I think I'll just stop right there. So thanks very much for your attention.
Well, thanks very much. That's certainly in keeping with some of the clinical data that we have post-stroke with neurostimulation in humans. Any comments or questions? I think we have mics on the sides. Yes, right in the back.
Hi, that's great data. I'm just curious how you, if you care to speculate about how you could use this kind of system in a clinical treatment, context.
Yeah, I mean, this is very preliminary. This is preclinical basic science work. I mean, I think one key thing would be to develop more red-shifted channel rhodopsins that could respond to longer wavelengths of light that would penetrate tissues further in a big brain rather than just a mouse brain. That would be one thing. Obviously, you'd need to inject a virus into a human to drive the expression of channel rhodopsin in these specific circuits. One thing that I like about this is that it was using a surface LED rather than driving optrodes or electrodes into the brain. And so if you have a powerful enough light with a red-shifted channel rhodopsin, it might be possible to effectively activate circuits in a thicker, more scattering human brain. But at this stage, don't know yet. I mean, we haven't improved recovery for stroke survivors, so I think we have to keep our options open. And
Great, beautiful work. So what you've done is a sort of a pathway-specific stimulation.
Yeah.
From the thalamus. Do you have any data on sort of a non-specific stimulus that could maybe mirror what would be applied clinically? So if you were going to use TMS or direct current stimulation, something translatable, are you really? I mean, it's elegant, it's beautiful. Do you know that that's required?
do I know if the cell circuit-specific activation is required? No, I don't yet. I mean, I suppose, I mean, we didn't, a priori, if we could tell if we hit the right nucleus in the thalamus by seeing the fluorescent expression in the intrinsically mapped cortical regions. So we use only ones that had the right injection location. I guess if we were to maybe go back and look at some of the, I'm not sure if my postdoc did all the analysis of the ones where we missed the injection. You're saying, like, say if we hit a broadly projecting thalamic nucleus that wasn't specifically associated with the sensory. Yeah, we haven't basically answered your question. We don't know if it's specific for the circuit, or if I was to hit, or if I was just to optically stimulate layer 2 neurons, would I get the exact same effect? I don't know, behaviorally.
Or could there be a way to indirectly activate the thalamus in that hemisphere? Let's say if you stimulated over the intact forelimb area in the other hemisphere.
Would I be able to drive thalamocortical responses? I mean, there's not really a lot of interhemispheric.
Thalamic responses in those same axons.
Anatomically speaking, I don't think you'd probably get that, because I mean, you'd have maybe some. There's not a lot of interhemispheric connections between the VPL and the two hemispheres, so it's more motor thalamic areas that have the interconnections. But yeah, I haven't done the study, so I don't know if that would work.
Dr. Buck.
Has any pharmacological intervention been tried that might just lower the threshold for activation in the brain?
Well, yeah, there's a lot of drug approaches that have, for example, you give somebody or give a rat caffeine or nicotine or some sort of stimulant, amphetamines the data is pretty mixed in the preclinical rat and mouse stuff with respect to stroke. So there is some data that supports this generally increasing the arousal could help with stroke, but it's not that consistent. So I don't know if that answers your question.
Well, thanks, everybody. Appreciate the topic. Thank you.